passa l'acqua? Ti lascio il bicchiere? Ok, qua siamo live. Allora, si inizia.
Please everyone have a seat. Now we are presenting Asia Mariani, the researcher of the University of Pisa. Okay, Asia, go on.
Okay, good morning everyone.
20 minutes and 5 minutes for the question.
Good morning everyone, my name is Asia Mariani and in this talk we'll see how light can be used to observe molecular vibration. First, I introduce the state-of-the-art technique to observe vibrational mode directed from the mean, but as the major disadvantage requires long acquisition time. Therefore, we'll see how we can manipulate the temporal profile of light pulses to reduce the duration of an experiment from several hours to just a few minutes, introducing a novel experimental skill. So, let's start. Let's consider a sample at equilibrium, meaning that all its molecules. This is not the right slide. Sorry. Okay. Again, let's consider a sample at equilibrium, meaning that all its molecules are in the electronic ground state. If we send on it a pulse that is broad in frequency, photons whose frequency matches the energy difference between the ground and first excited state are absorbed. So, if we take the intensity of this pulse that is called the probe spectrum before and after the interaction with the sample, we can record the absorption sample of the molecule under investigation. If we now send on If we now send on the sample of equilibrium a pulse whose energy matches that of the first electronic transition, again, a fraction of molecules are promoted to the first excited state. So, when the probe arrives now, it is a completely different configuration. First, it finds fewer molecules in the ground state that it can excite to the first excited state, and this phenomenon is called ground state breaching. Then, when it interacts with the molecules that are already in the first excited state, it can or simulate their emission back to the ground state, or promote them to a higher excited state, and this phenomenon is called excited state absorption. Now we can record the intensity of the probe spectrum after the interaction with the sample with the pump on and off to record this kind of spectra that are called alternate absorption spectrum. Here you see that the ground state bleaching and simulated emission lead to a positive contribution because this phenomena lead to more photons at this frequency compared to the case with the pump off. On the contrary, the excited absorption corresponds to a negative contribution because in this case photons are absorbed at frequency that are not absorbed with the pump off. So this quantity here depends on time, specifically on how much time passes between the arrival of the probe and the level of the pump pulse on the sample, or in other words, it depends on the pump probe delays. So we can record several spectra at several delays, for many pump probe delays, and how can we do that? By changing the delay of the pump probe spectrum by making the probe travel for a shorter or longer optical path. And then we can represent all the spectra collected in this kind of map that is a color map. How can we read this map? Here on the y-axis we have the wavelength of the probe pulse, while on the x-axis we have the pump probe delays. And each slice of this map, or each vertical slice, correspond to the signal at a fixed pump probe delay as a function of wavelength. So it is one single transient absorption spectrum. And you can see here that the color represents the intensity. So red represents a peak while the blue is a hole. On the contrary, if we take a horizontal slice of this map, we observe the signal at a fixed wavelength as a function of time. Okay, so far the only requirement we put on the pump pulse is that its duration is shorter than, that its energy matches that of the first electronic transition. But if we now use a pump that is short enough in time, we see some vibration on the map, some oscillations. You can see here in the map, or here even better if we consider a horizontal slice that again is the signal at a fixed time as a function, at a fixed wavelength as a function of time. And here you can observe the oscillations that are superimposed on the transient absorption spectrum. So what is the origin of these oscillations? Molecular vibrations. In a sample at equilibrium, molecules vibrate due to thermal noise. They all oscillate at the same frequency, but without a well-defined phase relation. A pump pulse whose duration is shorter than half the vibrational period forces the molecules to oscillate in unison, or, using another word, coherently. Mathematically, the pump pulse has created a superposition of the vibrational eigenstate that is not an eigenstate, so it evolves in time, and specifically this nuclear wave packet oscillates classically along the vibrational coordinate. And these oscillations modulate the optical properties of the sample, including absorption. So these oscillations here are the oscillations of the nuclear wave packet. If we now want to obtain the typical vibrational spectrum in the frequency domain, we must isolate these oscillations from the transient absorption baseline, and by removing the baseline, and then we Fourier transform them. Here you can observe several vibrational modes, because so far I've talked about a single mode for the sake of simplicity, but the real molecules have several Raman active modes. So first we can have a look at this spectrum. The molecule under investigation is a dye called crescent violet, and here you can see that it has a dominant peak at 590 centimeters -1, corresponding to the oscillations shown in this animation, then another peak at 520 centimeters, and three weaker modes at 490, 470, and 340 centimeters -1. So again, to obtain this spectrum, we perform a Fourier transform, and you know that when Fourier transforming an oscillation, we have to monitor the oscillation since it has essentially decay in order to not introduce an artificial broadening of the peak. Then, how finally should we measure this window? And again, this window is the pump probe delays window. How finally should we do that? According to the Niki's theorem, the sampling frequency should be at least twice that of the maximum frequency that we want to observe. So, for instance, let's go back to our measurement. Here we sampled a 3 picosecond long window with a sampling set of 10 femtoseconds for a total of 300 experimental points. So, for each experimental point, we have to acquire the signal of the probe spectrum after the interaction with the sample, with the pump off and the pump on. Then we change the delay and we repeat again this procedure, and we did that for 300 times. So you see that ISRS is I forgot to tell you that this technique is called impulsive simulated Raman scattering. This technique is a multi-step procedure. And here I wanted to give you a very simplified image to fix the idea behind ISRS. Let's consider a classroom of kids who are given the homework to take a picture of the same natural landscape at several sun positions. And to do that, they must follow 3 rules. First, they can take 1 picture per day. Second, they must all live at the same time to reach a natural landscape. And third, they must take the picture as soon as they reach the landscape. So every day the kids meet at the bus stop, then take the bus all together, go to the landscape, take a picture, and they have to repeat this procedure for as many days as many pictures they want to photograph. So again, this is a multi-step procedure. And going back to our experiment, what are the disadvantages of a multi-step procedure? So first, they lead to long acquisition times. that are disadvantages because they can reduce the signal to noise ratio, because the optical power of the laser, the power at the output again of the laser, fluctuates in time, is not constant. And ISRS signal, the vibrational oscillations, are highly sensitive to noise because, as you can see here, their amplitude is much smaller than that of the transient absorption signal, and this means that even small fluctuations of the background can overwhelm the oscillation. And second, multi-step procedure requires a long exposure of the sample to multiple light pulses, so we can't investigate with this technique specimens that are susceptible to photodegradation or irreversible processes. So, let's go back to our kids, and they could finish the assignment in just one day if each of them took a photograph of the same landscape at a different sun position. But how can they do that while still obeying to the second and third rule? Imagine that rather at the bus stop, the kids meet at a rental bike shop where they're giving a different vehicle based on their car. So red gets a racing motorbike, yellow a sporty scooter, green a much slower scooter, and for blue a kick scooter. So now they all start at the same time from the rental place, but since they travel at different velocity, they reaches the landscape at a different hour. So red almost immediately, followed by yellow, green, and after sunset by pure blue. So I guess you figure out the analogy that the color are the spectral components of the probe paths. Each day represents a delay between the pump and probe. And so now, what does the rental place represent? It represents a dispersive medium inserted in the optical path of the probe. Because when the velocity of a spectral component in a dispersive medium depends on its frequency. So if the two components start traveling in air, they travel at the same speed, and then at different velocities inside the medium. And as a result, when they exit the medium, they are delayed, they are located at different positions. If we have a look at this animation here, this is the propagation of a wave in a non-dispersive medium. You have to look at the red point. You see that all the frequency travel at the same speed, and as a result, the pulse preserves its shape during the whole propagation. In contrast, in a non-dispersive medium, let's wait that it starts again, still you have to look at the red dot. Now you will see that the frequency start together, but now the lower frequency travel faster than the higher frequency, and as a result the pulse stretches in time. The temporal dispersion between the different frequencies is described by a parameter called chirp, and as you can imagine, the thicker the dispersive medium, the larger the chirp. When we use a pulse in our experiment, each spectral component observes the oscillation of the nuclear wave packet at a different time. So red as soon as it is triggered, while blue when it's almost decayed. And the consequence of this fact, this is exactly the arrival time of this spectral component to the sample. Here T is the pump probe delay that we fix experimentally. Then C is the chirp that we introduced with the dispersive medium, and omega p is a frequency that we chose as a reference. And this is the equation written for the wavelength because we are going to use it in a minute. And the consequence of the fact that the time is now a function of wavelength is that the ISRS oscillation exhibits a slope in time. So if we now take a vertical slice of this map corresponding to the signal at a fixed pump probe delay as a function of wavelength, we observe oscillations in wavelength. And this is very different from what we did before when we took the signal as a function of time at a fixed wavelength. This was conventional in passive radiogram scattering. But now, in contrast, we are observing oscillations in wavelength at a fixed pump probe delay, but we know that time is a function of wavelength. So we can map these oscillations from wavelength to time. Now that we obtain again oscillations in time, we can Fourier transform them to obtain a spectrum. So, and here I just want to remind you that these oscillations are not present in ISRS when all the spectral component interacts with the sample at the same time. So here we are forced to scan the pump probe delays for many points by changing the delay experimentally, while here we can record the entire vibrational dynamics with only one measurement at a fixed pump probe delay. This is a simulation, and now let's have a look at a real measurement of this technique called chirp-based ISRS. These are the oscillations of, again, this molecule traced violet obtained by fixing the pump probe delays at 4 picoseconds, and again you can observe the oscillation superimposed on the transient absorption baseline. As before, we can subtract the baseline, isolate the oscillations in wavelength, then map this oscillation from wavelength to time, Fourier transform, and obtain the vibrational spectrum. And I want to stress that this is a real experimental spectrum that we acquired with a measurement of order of minutes rather than hours. But it turns out that much faster acquisition times is not the only advantage of chirp-based ISRS. If we take the signal at 2 fixed pump probe delays, which are slightly different, again we observe oscillations that are superimposed on the same transient absorption baseline, but oscillate out of phase. And why? Because the difference between these two delays is equal to half the vibrational period of one chosen mode, and in particular 28.3 is half the period of the most intense mode at 590 centimeters minus 1. If now we subtract these two signals, two things happen. First, the baseline is automatically removed. And second, as you can see here, the amplitude of the oscillation is increased, and as a consequence, also the amplitude of the peak corresponding to this mode is increased. And only the peak corresponding to the mode that we chose here to select our delays is enhanced, the other are not. So this means that we can use the pump probe delay as a knob to decide which mode see better than others, and this is very useful for the weak mode. So as an example, here we can observe a comparison between the intensity of the 340 centimeter minus 1 mode in a conventional ISRS experiment, where it is scarcely visible, and its intensity in a chirp-based ISRS experiment. So, to wrap up what we saw today, first we introduced transient absorption spectroscopy, a pump probe technique that allows the investigation of electronic excited state. Then we saw that with the same experimental scheme, we can observe also vibrational modes, provided that the duration of the pump pulse is shorter than half the vibrational period. ISRS is the state-of-the-art technique that enables the investigation of vibration modes directly in time domain, but requires long acquisition times. So to address this issue, we introduce a probe in a chirp in the probe pulse. When using a chirp probe pulse, each different spectral component interacts with the sample at a different times, and this enables to record the entire vibrational dynamics in a single step measurement. This means that we can not only achieve a much higher signal-to-noise ratio, but also investigate that samples that cannot be studied using conventional ISRS, that is, irreversible specimen and a sample susceptible to photodegradation. At the very end, we saw other two advantages of chirp-based ISRS, namely the automatic removal of the transient absorption baseline and the selective enhancement of chosen Raman modes. Chirp-based ISRS is a novel experimental scheme that I developed in the femtoscopy group at La Sapienza University of Rome during my master thesis and then in a year as a junior researcher. So I want to thank all the people of the group, and in particular my supervisor, Professor Tullio Scopigna, and Professor Giovanni Batignani, who is here today and who gave this talk yesterday. Then I want to thank the organizer of La Toscani for giving me the opportunity to present my work today, and all of you for your attention. Thank you.
that you have to vary the size basically of the dispersive medium. Do you actually vary the size, or is there another way to vary the the total dispersion? Basically?
We don't vary the size during the experiment. We just chose one that is.
No, yeah, but between when you have to understand how much dispersion do you need? Do you change the dispersive medium size, or is that?
Yes, maybe
Okay.
we can run some simulation.
Okay.
Not simulation, just simple calculation, because chirp is the second derivative of group velocity, so we can simple calculating the chirp, but then we characterize the duration of the pulse, because essentially, as I showed you before, the size of the chirp determine. I'm trying to find that slide. Sorry. Okay. Was it this one? Okay, sorry, I can't find the slides, but basically the size of the chirp determine the monitor spectral window. Okay.
Thank you.
I answer your question?
Yes.
You said there is a way to remove the baseline with the chirp-based method. In that case, you do have to take two measurements at different delays.
Yes, exactly.
Right.
If we want to do that, we must take two measurements. Why? If we do a single shot measurement, we must remove the baseline using the typical post-processing methods, like taking a smooth and then removing the baseline. But again, taking the measurement at just two delays is again still much faster than doing it for a conventional experiment. And it's also very interesting because we can choose the delay again to select which mode we want to observe better than the other.
Yeah.
Thank you, Asia, for your talk. And now I'll introduce you to the next student talker. So you have to know that in the last edition of Lights of Tuscany, a person kept appearing and helping us all the time. And that was Dorothea Nardini, the student that you see in front of you. She was there at every night call. She was there for every last email, for helping us building up the Florentine area of this event. And then at the last moment, she disappeared. Because she got into a summer school that was too good to say no. So she wasn't present for that day, and this is only fair that we asked her to come back as a special guest for her seminar today. She is a PhD student at the European Laboratory of Nonlinear Spectroscopy, and she's going to give you a talk on her research that's also correlated to cancer cells and brain cells. Thank you, Dorothea.
Thank you, Julia, for the nice introduction. Can you all hear me from here? Okay. So, good morning, everyone, and happy International Day of Light. So today I want to start with a question for you. What's the most important part of your body?
Brain.
Brain.
Heart.
Heart. Nice, interesting. So I'm a ballet dancer, so I might say maybe my feet. Someone who plays football might say the same. A chef could say their tongue. But I think that we can all agree with our next speaker that what really defines us as humans is our brain. This amazing organ is capable of so many things and is so important in all its parts because each one of them defines a side of you. Okay? So this is why it's so important to preserve it. And healthy brain tumor tissue preservation is a major concern in neurosurgery, where usually patients have to go through total tumor resection. So of course the best treatment would be for the surgeons to only cut the tumor and leave behind all the important tissue that define the patient as a human being. To do so, surgeons nowadays have different tools to help them define the position of the tumor. They are mainly in preoperative scans, so MRI, CT, and PET. They provide a really good contrast imaging of the tumor, and so a very precise location, but they have a problem. They are all preoperative. So this means that they are taken hours, but not even days, before the operation starts. And what happens is that in those hours, in those days, and especially at the beginning of the operation when the surgeons have to cut the skull, the brain unfortunately can shift a little bit. And this means that our preoperative scans are no longer 100% reliable. Of course, surgeons will still use them and try to compare them with what they see in front of their eyes in the brain of the patient. But they don't reflect perfectly the reality in front of the surgeon. So what I want to discuss with you today is if physics, and in particular spectroscopy, can help us define the tumor borders during surgery, so in real time. I work within a consortium of different universities that is called Hyperprobe, and our main goal is to develop an instrument that will help surgeons during surgery by imaging the exposed brain tissue and providing maps of the position of the tumor. It will do this based on hyperspectral imaging. This is a technique that combines the 2D spatial awareness of wide field imaging with the molecular information that we can retrieve from spectroscopy. So to understand better, I need to show you a scheme of our setup. Do you see better now? Then I turn it on again. So we have a plasma light source that emits in a very broad spectral range between infrared and near ultraviolet. Then the light is driven to the wavelength selector that sequentially selects different wavelengths between 385 and 1015 nanometers. We have 126 wavelengths now. And the light is shown onto the sample that for now is just a tumor biopsy. In the future maybe will be the skull of a patient. And the reflected light from the tumor biopsy is acquired by a camera, a CMOS camera. So basically the wavelength selector selects sequentially different light, and from each wavelength of illumination we get a single image. So in the end of one acquisition we will have 127 images that are of course in black and white, like grayscale. Here are colored just for showing purposes. And they will make up what we call an hypercube. An hypercube is a 3D matrix where the two of the axes are the axis of the camera, and the third dimension is the wavelength axis. So now I would like you to focus on just one single pixel in the imaging. So if you look at one single pixel, for each image you will have one value, the value of intensity registered by the camera. Okay? Taking all of these values, you can retrieve the raw reflectance spectrum. With some very boring and very easy math, we get from the reflectance spectrum to the absorption spectrum of that pixel. This is the absorption spectrum of the sample in that particular position that is mapped from that pixel. Of course, we can do this with all the other pixels, but to show you why this is important, I need to talk a little bit about molecules. So, of course, you know that molecules have fingerprints, just like us. Theirs are spectral fingerprints. And this is because each molecule, each kind of molecule, have a different energy level distribution, for those of you who already taken quantum mechanics. So they interact in a different way with light, with photons. And this reflects with different spectra of absorption of the light. Here I show you the main biomarkers that we are going to talk about. So we have hemoglobin, that is in blood. We have oxygenated and deoxygenated hemoglobin in orange and red here. Then we have water, of course, lipids, and various forms of cytochromes. Here I show you cytochrome deoxidase and reduced. Sorry guys, it's going on without me asking for it. And these cytochromes are related to cellular metabolism, so the activity of the cell. So when you look at the spectrum of our sample, you can look at it like a linear combination of all the spectra of the molecules that are inside the spectrum, the sample too. Maybe I turn on the light there so you don't fall asleep. Where this linear combination has coefficients, of course, and these coefficients are the concentrations of each molecule in the sample. So with some very nasty algorithms that are called spectral unmixing, we are able to retrieve the concentration of each of our biomarkers. So let's focus a little bit on the hemoglobins, so oxygenated and deoxygenated. In particular, let's focus on this region. Can you all see my pointer, right? Okay. In this region here, where oxygenated hemoglobin shows 2 peaks, while deoxygenated hemoglobin only shows 1 peak. So if I look at a tumor biopsy, either in real life or in an RGB image like this, I cannot say where the tissue is more oxygenated or where is less oxygenated, no? And it's not just me or you, even the doctor doesn't know how to say that. But if you acquire it with hyperprobe, and then you look at the spectrum in different points, for example, these 2 regions that I circled here, you can see the spectra of those 2 regions that are shown here on the right. So ROI1 shows 2 peaks in this region, while ROI2 shows only 1 peak. What does it say to us? Of course, it doesn't say that there is only oxyhemoglobin in region 1 and only deoxyhemoglobin in region 2. All of the molecules that we talked about will be in every point of our sample, okay? But it's saying that here, in region 1, we will have a lot of oxygenation, while in region 2, we will have more deoxidized hemoglobin. So doing so for each point of the sample, we can then color the image based on the quantity of oxygen present in the blood, basically creating a saturation map. And this is very interesting from the medical point of view because it gives us contrast and a very informative quantity, so the oxygenation of blood. This is something that is not directly accessible during surgery right now, because there are no tools that show you the oxygenation. In the future, when we will bring hyperprobe into the surgical room, we could be able to show an overlaid map of oxygenation over the patient's brain and help this analysis for the surgeons. So, of course, we did that for oxygenated and deoxygenated hemoglobin, but we can do that for many other molecules, such as cytochromes. Here you can see that all these yellow parts are more active, metabolically active than the others. And for example, also total hemoglobin that doesn't show anything about oxygenation, but tell us about where the blood is. Okay. So a crucial point in cancer treatment is the definition of the tumor grade, because of course both a surgeon and an oncologist will react differently during and after surgery if they are in front of a high-grade tumor rather than a low-grade tumor. So the standard practice is histopathology to understand the tumor grading. Histopathologists create images like this. So here we have two samples, the two columns, and two kind of histopathologic imaging. And you can see that even if we are not doctors, we can clearly see the difference. So we can say, okay, yes, if you tell me that when there is more pinkish regions, it's low grade, I can already tell you this is a low grade and this is a high grade, even if I'm not a doctor, okay? And both here, same here with brown and blue, okay? But when you see it with hyperprobe or with normal imaging, you cannot differentiate between these two samples. So why do we want to change? What's not good in histopathology? The lengthy procedure. Because sometimes it can take days, even weeks, to get these results. So we cannot do that during surgery, for example. So surgeons don't know the grade of the tumor when they start. They can guess, but they don't know actually the grade of the tumor. So what you can do with hyperprobe is to look at the spectra of those samples. So, for example, you can look at the spectra, and then with spectral mixing, we said that we can retrieve the concentration of main biomarkers. So you can take these two samples and then put them in, in a graph like this, based on the concentration of the molecules that you selected. And you can see that the sample on the right is here, while the sample on the left is here. Okay? And you could tell me, yes, but why would I do that? Why is it interesting? It is interesting because we analyzed a lot of samples. Each one of these points is one sample that we analyzed, and we displayed all in the same graph. And the magic happens when I turn on the colors, and I color them on the based on the grading that the doctor told me that tumor was. Of course, they told me some weeks after. Okay. So what's really interesting here is that the low-grade tumors, so 1 and 2, are clustering in the upper right region, while the rest of the graph is populated by high-grade tumors. So in a future, when we will have so many data set, a very big data set, we can define a threshold, maybe, something like this, that based on this threshold, we can have the spectrum of an incognitous tumor, and we can place it in this graph, and if it's lower than the threshold, we will say that it's like a high grade, otherwise it's a low grade. And this, once it's established the threshold, will be done in just a few minutes, because one acquisition is less than 30 seconds, and then the processing can be really quick. So, what I talked with you about today, the spectral mixing can provide us the concentration of the main biomarker, just starting from the spectra of our samples. Then, by the quantification of those concentrations, we are able, or we will be able in the future, to determine the grade and the type of tumor. And in the end, we also are able to create 2D very high contrast maps for the main biomolecules. But in the beginning I was talking about differentiating between tumor and healthy tissue, and I didn't talk about that yet. This is because clinical trials are hard and you don't start from the surgical room. And you also don't get biopsies of healthy tumor tissue. So for now, I never saw a healthy brain tissue. I never saw a healthy brain tissue. So the next step for Hyperprobe Consortium is the development of Hyperprobe 2.0, which will be the clinical prototype that we could bring inside the operating room. With this, we can image the entire exposed cortex of the patient during surgery. So in that moment, we will be able to have images of both healthy and tumoral tissue. And we could compare it, and we could start learning how to differentiate them through spectral mixing. So between what we did and what we will be doing, there is what we are doing now. That is experiments on living mice. This is a perfect bridge between tumor biopsies and what we will be doing in the surgical room. Because of course mice are alive, like patients, but on the other hand they allow us to use Hyperprobe 1 in the lab. So a very controlled environment, and also with a much bigger set of wavelengths. So I will show you just a little bit of this because we are under publication right now, so I cannot tell you much. But one of the experiments that we did with the mice, of course we put we put the head of the mouse under the Hyperprobe, and we used a mask to change the oxygenation levels in the air that the mouse was breathing. So these that you see here are different concentration maps of oxygenated hemoglobin with respect to the normoxia state, which is 21% oxygen, that is normal air that we all breathe every day. Okay, so that you can see at 40% you already have a little bit of an increase because 0 is very red and orange is over 0. And 60, 80 and 100% oxygenation show a very high oxygenation level of hemoglobin. While instead, starting from 21% and cutting the oxygen supply and going to 0% of oxygenation, we have a totally oxidized brain. So these experiments helped us showing that Hyperprobe is actually capable of monitoring the variation in concentration of molecules, here for example hemoglobin, during time and in real time. And this of course will be helpful to adapt it in the surgical room, because being quick is fundamental in that situation. So I would like to thank you all for the very kind attention, and I would like to thank the organizers that invited me. It's like being home back. And of course my research group and all the consortium. I'm open to your questions if you have any. Yeah?
How do you handle the fact that tumors may be inside of the brain, like deep in the tissues? Because you look at reflections.
Yeah. So
yes.
Basically, now we are doing tumor biopsies. So they are cut, and then after the cutting, some of them are given to the histopathologist, of course, and some of them are given to us. So we just see our sample. In the future, is just like the surgeons, they have to make their way inside the tumor. Of course, those tumors that are very deep into the brain are one are more difficult to treat, and also they are more difficult to treat because you have to cut a lot into the brain to get to them. So our target is of course more superficial tumors, but if the surgeons are able to create a window to reach the tumor, cut it, then also light can go through. Okay. Other questions? Yes?
Especially with the oxygen and blood saturation,
Yeah.
could you apply this not only to the brain but also to other body parts?
Yes,
Or
we are doing, actually we are doing some testing on the skin. We are collaborating with some hospitals for skin tumor analysis, and also other type of problems of the skin. And we are also starting to think about a collaboration with some biologists for the analysis of green, so like leaves and all this kind. Of course, hyperspectral imaging has a huge range of application because it's non-invasive. It's very cheap, like compared to other techniques, and it's wide field. So yes, there are a lot of applications also in art and many others. Okay, I don't know if there are any more questions. Okay. So okay. So thank you guys. Really enjoyed this very, very nice opportunity that you have been given, because I think it's amazing. They work a lot.
Okay, so it's time to introduce to you the third and last PhD candidate that we'll be talking today. And so a while ago we had heard about the opportunity to have a PhD candidate from Basel in Switzerland, and we were like, oh yes, if we can do this, it would be very cool. And we managed to do it, so it is my pleasure and my honor to introduce to you Nessim LoT.
Okay, this works. What an introduction, thank you. So, yes, so my name is Nessim. I'm a PhD student in Basel, in Switzerland. And today I would like to talk something a bit different than what you heard so far. My PhD project about how does DNA moves inside the nuclei. So I'll start very generally, if this works. No? No. Okay, so I'll start very basic. I don't know how much of this is trivial for you, but okay, so inside of our bodies and everywhere we have cells. And inside of cells, there are these things called nuclei in eukaryotic cells, so mammalian and everything. There's a nuclei in which DNA is contained. And this is in the order of 10 to the minus 4 meters. And, well, let's go back. Okay. And DNA is shaped as a double helix that is 10 to the minus 9 meters. And itself, it is composed of 4 bases that are A, T, G, C, that are molecules that are bound to each other by hydrogen bonds. That is the order of 10 to the 10 meters. So the first question is, okay, how much DNA do we have and how packed is it inside of the nuclei? Because the nuclei is a fixed shape, right? So we can get this very simple estimate. We can think of DNA as what we call an ideal chain because it's a long polymer. So we can do, okay, it's a random walk of n steps with fixed length L. And metric that we can compute for this chain is the average end-to-end distance. So how much space does the whole chain occupy? So for DNA, we know that we have about 2 meters worth of DNA. So if you put the bases back one on top of each other, you get around 2 meters. They are organized into chromosomes, these little filaments. This works. These little filaments that you see here. And the thing that we need to define is the length of this segment, and so how big the segments for DNA should be. This is what's called the Kuhn length, and this we could estimate it to be 10 to the minus 8 meters. So we have 10 to the 8 number of segments that are 10 to the minus 8 meters. So this, when you plug it into here, you get 10 to the minus 4 meters. So this is the amount of space the whole DNA inside of every cell or for one chromosome takes. But okay, I said the nuclei has a fixed shape. So what is the size of the nuclei? 10 to the minus 5 meter. So there is a problem. Doesn't work. So DNA has to be much more compact than just this random shape. And on top of this, there are 23 of these chromosomes per cells that are replicated, so you get to 36. So this then scales up completely. So it has to be very compact. That's the point I want to make. And okay, why does it have to be compact? So DNA is contains segments that are we called genes. These are pieces of DNA that are used to make proteins. Proteins are the things that make everything in the body work. And this process is very regulated because you have to think, I don't know, you cannot make a foot out of a cell at every time point. So the genes that control, very simplistically, the gene that control for making the foot has to be expressed at a very specific time points during the development. This is the kind of regulation I'm talking about. So, and on top of this, we found that, so these are genes, this is the DNA chain, and we found that many years ago, we found that there are pieces of DNA that are found very far from where the gene is, that are called regulatory elements, they're called enhancers, because we saw that when you remove them, something happened to the protein, and everything is dysregulated. And the cool thing is that when you look at how far they are from each other along the chain, these could be millions of base pairs away. So these are very far and somehow interact with each other to make proteins. So somehow there is regulatory information that is transmitted. And because we saw that DNA has to be very compact, it's very intuitive to think that this has to happen in space instead of along the chain has to happen in 3D space. And this has to be dynamic as well, because the gene cannot be expressed every time. So people found that there is this protein that I show here as a ring that is called cohesin that basically binds to DNA and then starts extruding loops. So as if you pull a string and then you look around and you pull on it, then you make a very nice loop. And this has been shown by microscopy that it works in vitro. But then the question is, okay, if you take two pieces of DNA separated by a given separation, how often does this process happen and for how long does it stay? So in my group, there's a postdoc called Mattia that started to say, okay, can we try to make a model for DNA that is not the random chain, that works, that recapitulates properties of DNA that I show, because then we could start to make prediction on what would happen for this process. This is what Mattia did. Here you see one of the simulation that we have, 2 beats, and the yellow part is the loop extruded by cohesin. So then the very cool thing is that once you have this model, you can say, okay, I take 2 beats, and around one of them, I draw this ball with a given radii, and I say everything that is below this radii is in contact with encounter. And then I can ask, okay, how long does this thing last when you compute the distance between the 2 beats, and how frequently they happen. So, how long do they actually last? So here what we can build is this type of histogram in which you have the probability that an encounter lasts at least tot seconds, and then you can do without this looping process and with, because you have the simulation. So what you see is that without, the probability decays very, very rapidly, because most of the encounters are just due to thermal fluctuation. So the 2 beats come close because they vibrate due to heat. But the cool thing is that when you add extrusion to the process, you start to get this long tail of very long-lasting encounters that are due to extrusion, that are very rare still, but very long. And how does this happen? So when cohesin binds in the middle of the 2 beats, it starts pulling on both ends, and then at some point, both of the beads will be at the bottom of the loop together, because the cohesin pulls each end at the same rate, and then both of them will be very close. And then they will stay for some time until they are inside the loop, and then inside the loop they fluctuate again. And then it's intuitive to think that this duration is due to the inverse of the speed of this process. The slower it is, the longer they will stay at the base. So okay, now comes the fun part. How do we actually experimentally test this process? So what does it mean to observe this process in using microscopy in this case? So we want to work with the live cells because this process happens inside of the cells, so we cannot fix them, cut them, put electrons on them, freeze them, all of these things, because they have to be alive and well, so at 37 degrees and yeah. And we have to look at the sample frequently, because this process happens very fast. As you see, it was in the order of tens of seconds, and for a long time. And for this, we need to use low amount of light. I will show you later why. And then, at the same time, we need to look at things that are in the order of nanometers, because the beads, the DNA fragments that I show you are in the order of the nanometer, are in the nanometer range. And on top of this, we are looking at things that don't emit light. So when you do fluorescent microscopy, you look at fluorescent emission. In this case, cells don't fluoresce by themselves, DNA doesn't. So you have to somehow introduce fluorescent molecules inside of the system. So I will show you how we do this. So why do we need, maybe I can turn off the light for this. Okay, why do I say that we need to use low amount of light? This is an example movie where, so dots represent protein, whatever that they are. And what you see is that, I hope you can appreciate that the cells are moving away, so they are dispersing towards there and there from here, which is where the light source is. Because cells are very sensitive to light, there's a lot of mechanisms regulating how cells handle light. Basically, they don't like it. So when you put light on cells, they will move away from it. And this is what happens here. And on top of this, something that has been talked about before, there is also bleaching of molecules. So fluorescent molecule at some point will stop fluorescing, and this is what you see, that the whole image gets darker. So we need to use very low amount of light. We need to be very gentle. The second thing was, okay, we look at things that are in the order of nanometers. Turn on the light again. And because we use light, we're limited by diffraction, as you probably know. And this is related to the wavelength and the aperture of the lens that we use. And usually for the microscope that we use, so for light microscope, this is around 200 nanometers. But the thing is, a protein is around 10 nanometers, so much smaller than this. And this is the scale that we would like to look at. So then the last thing is that we look at things that don't emit light. And for this, so in my project, what I did is that I took DNA, and then you can insert inside of the DNA sequences that you choose. And in this case, you can insert sequences that can be recognized by fluorescent proteins, so proteins that emit light when you shine light to them. And you can do this with the different colors, dyes as well, a lot of things. And then, so you can put two of them so that you construct two images in two different colors. And then how we actually image them. So we use this technique called high-low, in which instead of shining light directly to the sample, basically you shine light at an angle from the optical axis, so that you get like a beam of light that looks something like this. So that is transversal to the specimen. And this means that basically the amount of light that goes to the sample is very limited, because this beam, you can control its shape and its angle, so you can very finely tune how much light actually arrives to the sample. This is what we use. So basically in the end what we do is something like this. So this is a nuclei, and then there's two dots, because they are diffraction limited dots, in the two colors, and then we can measure the distance. And on top of this, what we can do to the cells is that we can remove the cohesin protein that we have, so we can tune how much extrusion, how much of this looping actually happens. So in the end, we do something like this. So this is an example movie in which you see the two dots it's a bit hard to see because they're small, but there you see the two dots oscillating. This is the distance between them and time. Okay, and then what we do is basically, because we know the shape that the spot should have, because it should have this airy pattern in 2D and 3D, we can basically localize the center of it using Gaussian fitting very simply, and then we can measure the distance between the two colors. And then basically our resolution then becomes how much error do we actually make while localizing the two and computing the distance. And actually, because we want to do something in which we have a distance over time, and then we put a threshold, and we look at how much is below this threshold. How much error do we actually have on the system is very important, because it will influence a lot the distribution of the things that are below the threshold. And so how we actually estimate this error in our images, basically we say, okay, instead of having two dots that are separated, that have a fixed distance that we don't know, what if we have only one, but then we observe it as we observe the two dots, so in the two colors? Then the distance between the two should be 0, if it's perfect. But actually, the distance with error becomes this Rayleigh distribution that we know the shape of, that has only one parameter. And actually, when we do the experiment, we can see that it actually fits quite nicely this prediction. And then you get an average error of around 40 nanometers. This is how you go from 200 nanometers down to the 40 nanometers, which is in the scale of proteins. So then, okay, this is what we want to measure in the end. So there is 40 nanometer error, but this is the event that we want to measure. So the question that you can ask is, what happens when you put error inside the simulation? The same type of error that we have in the experiment inside the simulation. So when the error is as big as the encounter radius that you choose, basically what happens is that you don't see anything anymore, because you're just sampling noise. But empirically, you can see that if the error is about half the encounter radius, you're still able to see these long-lasting encounters that were the things that we wanted to image in the first place. So with this in mind, basically the encounter radius that we can take is around 90 nanometer, because we have the error of 42. Yes. So this is actually experimental data now. In here I'm showing an example track that we recorded. This is the distance over time, and then we drew this line at 100 nanometer. And then we say, okay, what is the probability that an encounter happens for at least dot seconds? This is what happens when you have this looping, but then you can deplete it and see if the model was right. And indeed, when you deplete cohesion to the system, you get this, you lose these long-lasting encounters, which is what the model was predicted, meaning that really the fact of having this cohesin that make this loop brings together two pieces of DNA that are very far away from each other along the chain, but in space they can come together very closely for a long time, which we think is what drives then the regulation of expression. If you're interested more, I have more data to show this and how it links to the production of protein actually. And yes, as the last thing I wanted to show you, I wanted to show you that, I mean, what I showed you is just a part of the things that you can do with microscopy on cells. You can look at processes like this that are the replication of cells. So how does one cell becomes two, and how does the DNA is separated from the mother to the daughter cells? So this is something like this. You can also look at single molecule, so single proteins diffusing around inside the cell, and how far do they go, where do they, how much time do they stay bound, and stuff like this. You can also look at other type of replication, and here at the bottom this is transcription, so this is the production of protein happening in real time, and how does this relate to the encounters that I was telling you about. And this is already my last slide. So this is my lab. I would like to thank Luca, my professor, and yes, Mattia, with whom I shared the work, the microscopy facilities, that we have at the Institute. If you're interested in reading a lot more of biology, this is the preprint, our current preprint, which we did a lot more experiment. I'll be happy to talk about. Thank you very much for inviting me to this event. I hope it was interesting, and yeah, I'll be happy to answer your questions. Thank you.
I have a question.
Yes.
So you, in the beginning, you were saying that to have fluorescence, you change the sequence of DNA with some sequence that generate fluorescence, or if you attach the dye.
No, yes, we put sequences that can be recognized by something that has the dye fused to it.
Okay, and do you place those sequences in particular positions, or you're simply just trying to put somewhere and somewhere else and just saying, let's see how it's changing? Or do you put them next to something that is interesting?
Yes, so yeah, that's a great question. So, of course, everything that I told you really depends on where you are in the genome, because everything is very complex. So there's a lot of layers of things that I didn't talk about. So how we do it in the experiment, basically we have a fixed distance that is related to, on average, what is the distance between these two types of sequences, between the gene and its regulatory element. So this is 150 kilobases, which is, yes, on average what you get. And this is what we fix in the experiment. So we put one and then another one separated by this fixed distance.
Thank you.
Other questions? Everything is very noisy, but it's cool. Okay. Thank you.
Thank you to all the students who came here and helped us during this part of our event. Now I will ask you please to put your shirt on if you don't have it already. There is a button over here in the other hall also.